Hello, everybody. It is very good to see you once again. Compliments of the season. You're welcome to our endocrinology series. My name is Dr. Iya Izubasi, and we'll be taking mechanism of hormone action. So I am hoping that by the end of this class, you should be able to describe what hormone receptors are, their characteristics and functions. You should also be able to tell me something about the properties of hormone receptor interactions, the effect of hormone receptor binding. I would also hope that you should be able to describe the mechanism of action of hormones, the factors that affect target cell response, and the regulation of hormone secretion. Sit tight, it's going to be a bit long, very interesting. If you do have a question, remember to put the question in the target in our chat box, like our videos, and also subscribe to our channel for more videos. Remember that in the former videos, I had said that the hormones have a particular interesting characteristic, and that is that they bind specifically to specific cells and have specific action on those target cells to which they bind to. And that property is possible because of the ability to bind to what is called hormone receptors. So what are these hormone receptors? They are actually recognition molecules that are located on target cells. They're usually protein in nature and help to provide precise discrimination between the very low concentration of hormones we have in the ECF and other hormones or other molecules rather that circulate at very high concentrations in the same ECF. So usually they are classified according to the location of the receptor or according to the chemical nature of the hormone. And if we are looking at the classification of hormone receptors based on the location of the receptor, we do have cell surface receptors and intracellular receptors. The cell surface receptors are located on top of the cell, that is the plasma membrane, and because they are located there, they are referred to as cell surface receptors or plasma membrane receptors. It depends on the text you're reading. And they are usually for lipophobic molecules or hormones that include proteins, peptides, catecholamines, and eicosanoids. All right, because these molecules are lipophobic, they cannot cross the cellular membrane. All right, so they need to bind to the cell surface receptor. Binding to it now causes a kind of a generation of second messengers, which alter the activities of other molecules, usually enzyme within the cell, thereby, relaying the effect of the first messenger, which is the molecule. However, for the intracellular receptors, they are located inside the cell, and are usually for lipophilic hormones because those hormones can actually cross through the membrane into the cytoplasm and bind to either cytoplasmic receptors if they're in the cytoplasm or nuclear receptors if they are in the nucleus. And usually the classes of hormones that do this are the steroid hormones like your estrogen, progesterone, corticosteroids, your aldosterone, and the thyroid hormones. And binding to their cytoplasmic or nuclear receptors will cause an alteration of transcriptional activity of genes that are responsive to their action, thereby relaying the effect of the hormone in that cell. If you are classifying according to the chemical nature of hormones, we do have polypeptide and protein hormone receptors for lipophobic hormones, and then the steroid hormone receptors for lipophilic. All right, the polypeptide hormones or the protein hormones are classified into four: those that have CAMP as their second messenger, those that have CGMP as a second messenger, those that have phospholipidinositol calcium as a second messenger, and those who have kinase or their receptor has a kind of tyrosine kinase activity or a phosphatase activity, as a second messenger. So hormones that will have your CAMP as a second messenger include your adrenocorticoid hormone, your follicle stimulating hormone, as well as your luteinizing. Those that have CGMP as their second messengers include the atrial natriuretic factor. Those that have phospholipid inositol calcium as their second messenger include your thyroid releasing hormone, your gonadotropin releasing hormone, as well as your gastrin. Then those that have a kinase or phosphatase as their second messenger include your insulin, growth hormone, and your prolactin. The classes of hormone that will use the steroid hormone receptors will be, excuse me, the steroid hormones as well as your T3, T4 hormones. So, what are the function of these wonderful receptors? First of all, they enable the binding of the extracellular molecular hormone that is present in minute amount at the cell surface with very high affinity and specificity. Very, very important. Your affinity because of how low the concentration is in the ECF and specificity to be able to discriminate between molecules, all right? And then secondly, they relay the information that is inherent in these ligand binding effect to sites within the cell, not just binding. Binding is just one part. The second and most important part is to relay the effect of that binding to sites within the cell, whether it is alteration of enzyme activity, whether it is alteration of genetic activity, transcriptional activity, and all that. And then the third is to regulate hormone action by upregulating or downregulating the numbers of available receptors. So, having looked at receptor hormone binding, how do hormones work? So we are down to mechanism of hormone action. First of all, the hormone has to bind to specific hormone receptors before they can mediate the changes in their target cells. So when the receptor binding occurs and the associated cellular cascades also occur, these cellular cascades kind of like amplify the hormone signal, all right, allowing the hormones to act at very low concentration. Okay, so as a result of this, even though the hormones can circulate throughout the body and come in contact with a wide range of cells, they only affect cells that have the necessary receptors. If we have that in mind, let's look at this. So the hormone receptors, what things I want us to note about the target cells and their is that the hormone receptors help us to recognize, discriminate, whichever word you want to use, molecules with specific shapes and side groups. So you can see the square yellow binding to its own receptors. You can see the red one binding to its receptors. You can see this other cell has both red and black. So that's one of it. So it responds to only those hormones it can recognize. So for like this blue cell can recognize both the yellow hormone and the red hormone. You should also know that the same type of receptor may be located on cells in different body tissues, all right? Or the same cell, one cell can have receptors for different hormones. Also, the cells, the hormonal cells have the ability to be, the hormonal receptors rather have the ability to be upregulated or downregulated depending on the need for it or the concentration of the circulating. You should also know that the same type of hormone may have different effects in different body tissues, or the same cell with different receptors can have receptors for different, all right? Then if we have looked at that, we should also look at the properties of hormone receptor interactions. Number one, the hormone receptor interaction we should know is rapid and reversible, which is why the interaction can also be terminated very rapidly for control of hormone activity. Also, the receptor specific, there's receptor specificity. Insulin can only bind to insulin receptors. Thyroid hormones can only bind to thyroid hormone receptors. Thirdly, there is receptor affinity. This is why the cell is able to achieve significant binding at physiological level. So the affinity can be adjusted either by upregulation or downregulation of the receptors available. Then you have saturation. There is a finite number of receptors on the target cells. When those are all bound, they become saturated. All right. Then some hormones do have agonist and antagonist effects. So molecules that elicit the same biological response as naturally occurring hormones are termed as agonists, and those that bind but don't are called competitive antagonists. So what are the effects of hormone receptor binding? So once the target cell receives that hormone signal, it can respond in a variety of ways. You could have stimulation of protein synthesis, activation or deactivation of enzymes, as the case may be. You can have alteration in the permeability of cell membrane. You could have altered rates of mitosis and cell growth, and stimulation of secretion of products. All right. And you should also note that a single hormone may be capable of inducing different types of responses in a given cell. Yes, it is possible. So, what are the pathways involving intracellular receptors? The different receptors have different mechanisms of action. So, we would look first of all at the pathways that involve intracellular hormone receptors. First of all, like I've said before, that because the steroid hormones are kind of like lipophilic, they can easily diffuse through the cell membrane of the lipid layer to reach their intracellular receptors located in the cytoplasm and in the nucleus. All right. And we have said, and I reiterate, that hormones that can do this include your steroid hormones and also your thyroid hormones, because they do have these benzene rings with iodine, which are also lipid soluble and therefore can enter cells. All right. So what happens is that a steroid hormone will bind to its receptors in the nucleus or in the cytoplasm, and in this case, this binding can result into the formation of a hormone receptor complex, which now moves towards the chromatin in the cell nucleus and binds to a specific segment of the cell's DNA. Then the gene is now transcribed, produces a messenger RNA, which is then translated into the desired protein within the cytoplasm. For thyroid hormones, usually they usually bind to hormones that are already bound to DNA, unlike the that had to bind to the receptor, then move to the nucleus. The thyroid hormones go directly and bind to receptors that are already bound to DNA. And the binding of the hormone receptor complex to DNA now initiates your transcription of a target gene to mRNA, messenger RNA, which now moves to the cytosol and directs protein synthesis by ribosomes. So this is a pictorial form of all that I have said. You can see the lipid-soluble molecule going through the lipid bilayer, the capillary tube, okay, into the ECF, crossing the cytoplasm, okay, into here. So once it diffuses, the hormone diffuses through the plasma membrane, it now binds to a receptor. You can see that. And when the hormone binds with the receptor in the cytoplasm, it forms what is called a receptor hormone complex. This now moves into the nucleus and triggers gene transcription, the DNA. Okay, that gene transcription is now transcribed into an mRNA, which now is translated into proteins that can alter cell activity, right? You can see the activated protein. So that is for pathways that involve intracellular hormone receptors. On the other hand, that involving the cell membrane receptors is quite different from that involving intracellular receptors. For the cell membrane hormone receptors, the hydrophilic or water-soluble hormones must communicate with a receptor on the cell surface. Why? because they are lipophobic. And because of that lipophobic nature, they cannot permeate across the lipid bilayer of the cell membrane. And all amino acid derived hormones actually bind to cell membrane receptors that are situated, at least in part, on the extracellular surface of the cell membrane, with the exception of thyroid hormones, which I've already explained because of their benzene ring and iodine, they are lipid soluble, so they can't cross. Okay, so as a result of their binding, they do not, because they cannot cross over to bind to cytoplasmic or nuclear receptors, they cannot directly affect target gene transcription. But what they do is to initiate kind of signaling cascade, different cascades resulting into the amplification of the signal, and is carried out by a molecule called a second messenger. And the hormone, in this case, is referred to as the first messenger. Let's look at a pictorial description of this. You can see the water-soluble hormone passing from the capillary into the extracellular fluid. Because of its nature, it cannot permeate the cell membrane. So what it does is to bind with a cell membrane receptor, and the binding of the cell membrane receptor activates a G protein, and the activated G protein now kind of like activates adenyl cyclase. All right, adenyl cyclase catalyzes the conversion of ATP to CAMP, which is the secondary messenger in this particular pathway. Then CAMP now activates a protein kinase, and the protein kinase, after being activated, phosphorylates protein in the cytoplasm, and this activates proteins, allowing them to alter cellular activity. So we can see the innate differences between this and this. All right. So we should note that not all water-soluble initiate CAMP. Some others, like I've said, use CGMP, calcium ions, DAG, inositol triphosphate, or the protein tyrokinase, tyrosine kinase activity as second messengers. So, having looked at that, what are the factors that affect target cell response? The first thing is, what is the amount of available binding? If you do have a high levels of hormones, then you should have a high target cell response. If you have low levels of hormones, you should have low target cell response. If you do have normal, you should have normal target cell response. So the amount of available for binding determines the target cell response. Then if there is up or down regulation of the number of receptors, located on the target cells, or desensitization of the receptors, target cell response may be affected. And also, there are cases where you have two or more hormones working together or working against each other that can affect target cell response. For instance, if you have, let's say, the presence of one hormone which enables another hormone to act, you have a kind of permissive effect, two hormones working together to, kind of elicit a permissive effect where presence of one hormone enables another hormone to act. For instance, you have the presence of thyroid hormone enabling some productive hormones to act. That is why if thyroid hormone is low, in some cases, they have infertility in some people. This is what is responsible. Then you could have two hormones working together to elicit a synergistic effect. In this case, the two hormones with similar effect produce an amplified response. For instance, FSH working together with estrogen, all right, can help us have an amplified response. And then the antagonistic effect, where you have two hormones having opposing effects. All right. A familiar example is insulin and glucagon. Insulin is a hypoglycemic agent. Glucagon is a hyperglycemic agent. So when glucose level is high, insulin is produced, you understand? But glucagon has a kind of antagonistic effect on it, opposite effect. So those are the things that can affect target cell response. So how is hormone secretion regulated? Hormone levels must be tightly regulated in order to avoid an aberrant hormone level, a hypostate, a hyperstate, and both of these kind of states producing a potential disease state. Whether it is too high, it's too low, both of them are capable of producing a disease state. So the balance between the hormone production and degeneration allows body to retain this regulation. Okay, so initiation and maintenance of the majority of the hormone secretion in response to the different stimuli are governed by feedback loops. Like I said, feedback, feedback, feedback. What is feedback? Kind of a response to an information that is given. So we do have negative feedbacks and positive feedback. I will give you a classical example. Negative feedback. Here we have hypothalamus, pituitary, thyroid gland axis, and is usually controlled by negative feedback. Usually in normal situation, you will have the hypothalamus in the neurons secreting the TRH, your thyroid releasing hormone, that results in stimulation of the anterior pituitary to release TSH, which is the thyroid stimulating hormone, which will now act on the thyroid hormone to increase T3 and T4 secretions, thereby increasing the body T3 and T4 concentrations. When these T3, T4 concentration levels increase in the blood, okay, a signal is sent back that inhibits the secretion of TSH and TRH. And in the end, you have reduced secretion of T3, T4. This is kind of like negative feedback. High levels of your target gland hormones inhibiting your stimulating hormones, inhibiting your thyroid releasing hormones, and in the end, if these stimulating and these releasing and stimulating hormones are reduced, consequently, the effect on the target gland will also be reduced, leading to reduced T3 and T4 levels. Do you understand? If you do not understand, please ask a question in the chat box. I'll get back to you. But if you do, please like our video and subscribe to our channel. So, we do have another case where we have both negative and positive feedback mechanisms working, especially in the menstrual cycle. Most of the time in the menstrual cycle, we do have negative feedback working, like we saw in the hypothalamus, pituitary, thyroid hormone axis, thyroid gland axis. But when it does come to day 14 to 12, the 12 to 14, which which matches with our ovulation period, we do have a positive feedback working. So usually in the negative feedback, we have rising target hormone levels signaling the hypothalamus and pituitary to reduce hormone production. But when it gets to the ovulation time, do have rising hormone levels, giving feedback to the hypothalamus and pituitary to increase hormone production in positive feedback. So usually during days 12 to 14, which corresponds to our ovulation days, increase in estrogen level actually stimulates the increase in LH level. So you have what is called an LH surge, which will cause more increase in this, cause more increase in that, until the that's the mature follicle is released and ovulation occurs. So that is a very, very, very important example of a positive feedback. Or usually in nature, most of the feedback control in the body is usually the negative feedback control. So if you do, having come to the end of this lecture, I want to ask, do you have any questions? If you do have any questions, please put it in the chat box I'll get back to you. Thank you very much for your time. Bye bye. Until we get to you next class. Thank you for your time and God bless. Bye.